Hello, everyone, and thank you for joining me today. My name is Dr. Clayton Magill, and I'll be taking you on a coalification adventure as we discuss alkyl lipid influences on methane sorption in coals. The basis of today's discussion is actually quite fundamental. We'll be thinking about how coal, specifically its molecular structure, can affect gas absorption in both a very small scale sense, but also in an upscaled coal seam sense. Essentially, what we'll be looking at is how the molecular components of coal facilitate, or the opposite thereof, the absorption of different gases into that coal rock. We'll be focusing in particular on methane today, but this same concept should apply to a variety of nonpolar and volatile gases. To begin our conversation, I want us to think about the main influences on coal structure, at least from a geologic standpoint. If we think about coal rank, we're essentially talking about the maturation process. As we go from very immature or low rank coals like lignite, we find that they have vitrinite reflectances of less than 0.5. As we get to subsequently higher ranks, more mature coals, we see our bituminous and anthracitic coals, with anthracite coals having vitrinite reflectance values of above 1.9. In parallel with these changes in coal rank and maturity, we also see changes in macromolecular structure. As we go from top to bottom, we see that lignite and other immature coals have quite complex, at least in a relative sense, molecular structures where they have lots of oxygen, nitrogen, and sulfur atoms as represented by red, yellow, and blue bits on the macromolecular structure figure, as I'm highlighting with the laser pointer now. As we then get more and more mature, we see that a lot of these functionality groups go away, but also some of the complexity of the aromatic rings go away. They lose their double bonds. We can then take these macromolecular structures and fold them based on chemical principles into a unit cell structure outlined in the blue box here, and think about how that blue box will then interact with gases like methane, both in terms of the gas they can trap, as well as the gases that can absorb to them. In order to start this discussion, we're going to also think about a very specific set of coal samples. We have one set of coals derived from Germany during the Carboniferous, 7 samples that span vitrinite reflectance values from about 0.9 to 2.35, and we have some Permian coals from China that span a slightly larger vitrinite reflectance range from around 1.7 to around 3.5. All in all, a fairly large and overlapping range in our vitrinite reflectance and therefore rank or maturity of our coals. The other nice thing about these data sets is we can compare them to some of the previous data that's been published about them, at least in a bulk sense. Although today we'll be talking mostly about molecular signatures, in a bulk data sense, we can see that the new samples we're looking at, or the samples we're looking at, follow very well along the data published by Bush, Hahn, and Magill in 2019, where we see that the percent oxygen functional groups shows a decreasing trend with increased vitrinite reflectance and therefore rank or maturity. In a similar sense, when we compare the percent oxygen to our Langmuir volume, this is going to be the space available in our coal for gas absorption, we see that lower amounts of percent oxygen seem to show an increased amount of Langmuir volume, leading to the idea that perhaps the loss of some of these functional groups is opening up space. With that in mind, we can start to explore the new molecular data for the samples used in this study. These are essentially chromatograms, meaning GCMS data, for the extractable lipids out of these coals. If we look in the top left corner of the current slide, what you'll see is the relative abundance of different alkyl lipids. These are going to be our long-chain hydrocarbons as well as alkenes, alkenes, and if there's anything there, alkynes. And it's traced using the MZ85 component of our GCMS chromatogram. Each one of the peaks we see represents a different aliphatic component of our sample. We've done the same thing then for the extractable components that are oxygen functional group bearing, that is alcohols, acids, ketones, etc. And we've derivatized those with a trimethylsilyl or TMS group to make them more easily detectable with the GCMS system. And then we're showing you those compounds using the MZ73 trace. So each one of the pieces you see here is an oxygen-containing functional group compound. At a very first blush, we see that there are some substantial differences between the Carboniferous and Permian coals, namely that we have a longer average chain length for the Carboniferous coals as compared to the Permian coals. And it seems that overall, we have a slightly larger amount of oxygen-containing functional groups, at least extractable oxygen-containing functional groups, in the Carboniferous coals as contained in the Permian coals. Note that this does not necessarily mesh with the bulk organic data that we saw a few slides ago, which is interesting in its own right. We can then take the relative data gleaned from the last slide and actually compare the amount of extractable hydrocarbon content, basically integrate underneath the chromatograms we just looked at, and compare that to the vitrinite reflectance for those respective samples. What we'll see is that as the vitrinite reflectance of the sample, the maturity or rank of the sample goes up, we tend to have larger amounts of extractable hydrocarbons, whether in the Carboniferous or the Permian samples, although the two sample sets show different behavior. If we do the same thing for the extractable oxygenated functional groups, we once again see a positive relationship or upwards relationship between the amount of extractable oxygenated functional groups in micrograms per gram dry weight as compared to vitrinite reflectance. Although the variability explained by this relationship is quite low as compared to our extractable hydrocarbons versus rank. We can also take the same integration peak area data and compare them to age rather than rank. We again see some very strong relationships between hydrocarbon content and age, whereby we again see positive relationships between both the Permian and the Carboniferous samples, although there is disjointed behavior once again. And we see a slightly different relationship, albeit a slightly stronger one, between the oxygenated functional groups and age, whereby the older a sample is, the less oxygen-containing functional groups that we see in them. However, what I want to do next is actually take the ratio of these oxygenated versus hydrocarbon lipids and compare them to rank and age. If we do rank again, we see two very strong relationships that are almost parallel between the Carboniferous and the Permian samples, whereby the lower the oxygen to hydrocarbon ratio, that means not just the amount of oxygen bearing functional groups or hydrocarbon lipids, but the ratio of the two, the lower that ratio, the more mature the samples tend to be. And if we then compare that same ratio to age, we start to see the entire nature of the graph snap into a different focus, where we have a more consistent behavior across all of our samples and all of our ages. In fact, we can actually put a single regression line on this plot that has a R-squared value or a Pearson correlation coefficient value of above 0.95, meaning that 95% of the variability that we see in our extractable oxygenated to hydrocarbon lipid ratio can be explained by age alone. It's a very interesting relationship because we can then expand that to think about our Langmuir volume, wherein we see that the more or the higher the oxygen to hydrocarbon ratio is, the higher the Langmuir volume or the available space for basically gases to absorb occurs as well. Again, suggesting that the abundance, but also the orientation or the interaction between oxygen-containing functional groups and hydrocarbon lipids is an important control on coal structure, especially with relationship to gas absorption. We've identified a potential mechanism here that we can talk about, whereby we can take the organic matter, the bulk organic matter that's not extractable, the minerals and absorptive gases, and show them with the yellow, and we can put them in sort of a matrix of hydrocarbons and oxygen-bearing groups. As we start to break that matrix, meaning that we take or add hydrocarbons, but at a slightly different rate that we take or add oxygen-bearing groups, we can start to alter that structure both in 2 and 3 dimensions so that the organic matter, including gases, has an easier or I guess, alternatively more difficult time getting into or out of the macromolecular structure of the gas, of the coal itself. And as we go through time, we will lose disproportional amounts of the oxygen-bearing groups or the lipids themselves. This is really interesting because the other thing that changes a lot through the Carboniferous to Permian transition is plant taxonomy. At that point, we start to get a lot of plants that actually build three-dimensional structures with waxes on their lipids to protect themselves from sunlight, bacterial invasion, and a few other bits and pieces. But basically, they protect themselves with these so-called leaf or epicuticular waxes. Over time, the carbohydrate-rich structure that you can see here, this is a little slit called a stomata that's surrounded in white by the leaf waxes. The leaf wax structures are actually preserved even though the inside part of the leaf disappears, leaving a very interesting three-dimensional pockmarked structure like you can see in the inset of A here. What's also interesting is that same transition in leaf waxes is associated with major evolutionary changes in plants, most notably a massive drawdown of a lot of lycophytes. So these are going to be plants that grow almost exclusively in swamps at the expense or at the benefit of ferns and conifers, so sort of gymnosperms, Christmas tree looking things. And that change in plant taxonomy is associated with age, and therefore we can start to maybe see the influence of plant evolution as well as the chemical changes associated with that plant evolution influence coal behavior via its structure. In conclusion, we argue that the ratio, as opposed to the absolute abundance of different lipid types, correlates with age more so than rank, which suggests then a kinetic and or taxonomic control on the structure and absorptive behavior of coals. The extractable lipids that we're looking at in this study might influence methane absorption via their influence on macromolecular structure, how they interact with the residual bulk of coals. And we argue that future studies are important and should compare extractable lipid signals with analyses of bulk or residual coal data, such as ultimate proximate characterization and FTIR, so that we have an idea of the extractable and the non-extractable components associated with this sample. With that, I want to thank you for your time and make myself and my co-authors available for any questions or comments.